Govinda, Adarva Inamdar, Olivia Lidwin, David Taufilis, Megan Zhang, Vaishnavi Pati, and Daisy Yap.
First, let's begin with a brief introduction. Glioblastoma multiforme is the most common primary brain tumor found in adults. The prognosis for people with glioblastoma is quite poor, with 75% of adults dying within the first 18 months of diagnosis. This isn't so much the cancer in its initial form, but rather its great propensity to metastasize. As you can see in the scan on the slide, the tumor metastasizes to different parts of the brain. Tumor-associated macrophages, also called TAMs, are very crucial in this metastatic process. Osteopontin perhaps plays a role in the cytoskeletal movement of TAMs.
To further explain this pathway, as shown in the diagram above, we believe that glioblastoma produces a ligand that binds to a receptor in the tumor-associated macrophage. And because of this, we feel that this is responsible for the uptake and production of osteopontin, which is a gene that is known to have effects on the metastasis of glioblastoma. And because of this, there's also able to be tumor cytoskeletal movement within the tumor-associated macrophage. And this is able to move closer to the tumor microenvironment, and because of that, better influence the metastasis of glioblastoma. So when it's in this close proximity, we believe that there is an initial ligand that is formed to influence that metastasis. So because of this theoretical pathway, we were able to develop our hypothesis that there will be an uptick in the expression of osteopontin in tumor-associated macrophages, which could potentially play a role in the metastasis of glioblastoma.
Now, let's take a look at the experimental methods we used to collect our data. First, we used quantitative PCR testing to determine whether osteopontin expression was in fact higher in tumor-associated macrophages as compared to untreated macrophages. To actually measure this expression, we used some gene-specific primers, including osteopontin-specific and GAPDH-specific primers, including cybergroup dye. We also measured the fold changes for the various macrophages, and for later reference, a higher fold change value indicates higher osteopontin expression. So we compared the fold change after 4 hours of qPCR for the untreated macrophages and the glioma conditioned media stimulated macrophages, which glioma conditioned media basically just tumor juice. And then we compared the fold change after 24 hours for the untreated macrophages and the glioma conditioned media treated macrophages. And we did this comparison for M0 unstimulated macrophages and M1 and M2 stimulated macrophages. And as you can just see on this image here, this is the process by which we actually did quantitative PCR analysis.
To determine osteopontin expression through confocal microscopy, we use fluorescent staining. 6 microphage samples were stained for osteopontin in total, 2 of which consisted of unstimulated macrophages, and 4 of which consisted of macrophages stimulated in glioma media. To stain these cells, we added primary and secondary antibody containing chlorophore to detect the osteopontin. We then counterstained the macrophages using various fluorescent dyes such as SNAPI to stain the nuclei, GFP to achieve high resolution, and AF647 to stain the osteopontin. Then we measured the fluorescent intensity for each sample through an image processing program called Image Data Collect Quantitative Data. We specifically assigned and selected for GAPI, GFP, and the AF647 to be separate program channels to create an overlapping image of the entire macrophage, the nuclei, as well as the osteopontin expression, which allowed us to find the mean osteopontin in each sample.
Here are the results of our project. For our first qPCR, the fold change, which measures the amount of osteopontin in each type of macrophage, can be found in this table. As shown in this graph, there was a 53% decrease, about a slice and a half in osteopontin from the M0 macrophages to the M1 macrophages. Next, there was a 108% increase, almost a double in osteopontin expression from the M0 macrophages to the M2 macrophages. Additionally, M2 macrophages expressed about 4.4 times more osteopontin than the M1s. Therefore, M2 macrophages express the highest amount of osteopontin. Moving on to our next qPCR. The fold changes for the 4-hour and 24-hour untreated and treated macrophages can be found in this table here. As shown in this graph, for the 4-hour macrophages, there was a slight decrease of 12% in osteopontin expression from the untreated macrophages to the treated macrophages. And for the 24-hour macrophages, there was a whopping increase of 173% in osteopoietin expression from the untreated to the treated ones. Therefore, there was an unexpected decrease in osteopoietin expression in the 4-hour macrophages and a significant increase in the 24-hour ones.
So these are the results from our image day analysis. This is using OPN fluorescence, osteopoietin fluorescence, to quantify how much OPN is expressed in our macrophages. Over here we have 2 images from our image day samples. We did 8 total, but I'm just showing you 2 here. On the left we have our untreated macrophages, and on the right we have our treated. So these green blobs would be the macrophages. So we're looking at the intensity of the fluorescence signal from these macrophages, and we're comparing them between untreated and treated. So on the right, we have 2 charts. They're both the same. This is a data table, this is a bar chart. But basically, we're just showing average OPN fluorescence intensity, and that has about a 1.56% increase from untreated to treated.
There are several observations and conclusions that we can make from our results. For one, the results support our hypothesis that osteopoietin is expressed in higher amounts in tumor-associated, or M2 macrophages. This can be seen when comparing the osteopoietin expression between M1 and M2 macrophages. Additionally, the 4-hour stimulation show a decrease in osteopoietin expression, which is contrary to the expectation that it would increase based on previous research. Therefore, further studies can be done on the effect of different stimulation times on osteopoietin expression after GOCM stimulation. And lastly, the conclusion that the upregulation of osteopoietin causes M1 macrophages to switch to M2 macrophages has great clinical significance, as it may help scientists discover new methods to prevent and possibly reverse the effects of tumorigenesis.
Okay, so a couple of quick reflections on the process. A big thing that this project accomplished was it corroborates existing research, specifically a study by the Journal of Clinical Investigation, which also used similar techniques such as immunoblooding, ELISA, and qPCR. The process could have been improved with repeated trials, furthermore, we ultimately were unable to use CRISPR-Cas9, so 2 things that could have been improved in future trials. But really big outcome of this is that it gives a scientific basis for targeting osteopontin proteins via aptamers, which are selectively binding single-stranded RNA molecules, which have the effect of basically nullifying those osteopontin proteins, preventing downstream effects, and stopping that M1 to M2 switch, keeping these macrophages and T cells in their active cancer killing stage.
All right, so undoubtedly osteopontin expression is significantly higher in tumor-associated macrophages, leading to the metastasis of certain tumors, in our case glioblastoma. Osteopontin has an aggressive pro-migratory nature, leading to the metastasis of such related tumor cells. To expand on what was mentioned earlier, we were unable to use CRISPR-Cas9 because we wanted to actually see the results of knocking out the osteopontin in such tumor-associated macrophages, but we're unable to due to limited resources. We also were not able to run multiple trials, less guaranteed, and it was also important to mention that due to us doing the research in person in the lab, our methodology could have been skewed due to human error. Lastly, regarding future experiments. So regarding future experiments, we do believe that in a hypothetical treatment scenario where we try to lower general levels of osteopontin in the human body could lead to severe health effects, as osteopontin also has beneficial benefits to bone health and other factors. So that's important to mention in regards to how we can use this research to treat related tumors.
Overall, our research corresponds with and complements previous research that's already been done regarding OPN expression in glioblastoma. Beyond that, our research goes a step further and notices that using qPCR and fluorescent imaging that there's an initial dip in osteopoietin expression upon GLCM stimulation. Our results overall open up the door for future therapeutics that could target OPN proteins to try to treat glioblastoma. Before concluding, we would like to thank Dr. Coniglo and King University for providing us with the environment and the resources necessary for this study. We would also like to thank our fantastic team lead and assistant team lead, Angela and Sofia, for providing us with the directions and leadership necessary for this study. We would also like to thank the Governor's STEM Scholars, their sponsors, as well as their amazing director and assistant director, Ari and Elise. We would also like to thank our esteemed judges for providing us with the opportunity to present, as well as for their time. I would also like to thank you, and more broadly, everybody in the audience for giving us your undivided attention and allowing us to explain our project to you. Thank you.